I show you pictures that are on the test. So let me show you the pictures of what's on this test. Yeah, there's the first picture. Basically, you have to know 1 through 6 and what it means. So this is showing you endochondral ossification. That's right there. That should mean something to you. It means it starts with a hyaline framework, hyaline cartilage framework. This is your primary ossification center. That's your secondary ossification center. 2 is your epiphysis. All of this is your diaphysis. 1 is showing you the hyaline cartilage that's still there. 3 is actually still hyaline cartilage. It's your growth plate. That doesn't get ossified until you're 18 to 24 years old, depending on if you're male or female. So this is showing you endochondral ossification. And if you know what one, and what's for? Okay, whoever said perichondrium, I would agree if this hadn't been ossified yet. But it's periosteum, right? Because this is, right here it's been ossified. So it's periosteum. It was perichondrium when this was hyaline cartilage. So yeah, this picture. Go ahead.
Why were you looking for diaphysis or like bone?
I'd have to look at what the answers are, but it could be diaphysis or it could be bone. It's no longer cartilage, now it's bone.
Would you call it cortex?
No, I would not. No.
It's not the outside.
Well,
I can't tell.
I really can't tell if it's the osteon or not, but I either call it bone. It's not the medullary bone, that's for sure, so. All right, so that picture's on here. Ready for the next picture? Picture's on here. Let me make that picture big. So this is muscle. So again, 1 through 9 it looks like. 1 is mitochondria. 2 looks like the sarcolemma. 3, what do you guys call 3? Myofibril. The myofilaments are the thin and thick filaments. This is showing you the entire myofibril. And then you can see that 4 is pointing towards the thin filament, and 5 is pointing towards the thick filament. And so 4 is pointing to the troponin, tropomyosin, and actin, that's the thin filament. 5 is pointing towards the myosin, 300 or so myosin molecules that make up the thick filament. 6 is pointing towards the sarcoplasmic reticulum, which contains calcium. That's on the test. What stores the calcium that gets released from the action potential? SR, sarcoplasmic reticulum. 7 is pointing towards the T-tubule. 8 is pointing towards, I have no idea. What the heck is 8 pointing towards? Oh, no, I don't know. Oh yeah, I do. No, I really don't. Well, do you or don't you? All right, really don't know where 8 is pointing towards.
Which one? 8 over the right bottom. That's what I thought.
Oh, it's just cytoplasm, sarcoplasm. Okay. All right. No wonder I don't know what it's pointing to. It's unimportant. 9 is pointing towards part of a triad. 9 is pointing towards the terminal cisternae of SR, which remember, this is a triad. Terminal cisternae, T-tubule, terminal cisternae. This, not part of this picture, but there's another question about a triad on a test. So this picture is on the test. Now remember, the question might not say, what does number 8 represent? The question might say, which number stores calcium? Which number propagates the action potential to the inside of the cell? You guys want to know the answers to that? Okay, the number that stores calcium is SR. So 9 could actually be arguably an answer to what stores calcium. It's the terminal cisternae of the SR. And 6 could be an answer to what stores calcium, because it's SR. Hopefully I don't make you choose between 6 and 9, or hopefully 6 and 9 are answers, something like that. But Don't think that the question's gonna be as simple as what's number 9, it's not. It'll make you think more in depth about that. Any questions about this picture? Alright, next picture. By the way, you guys want me to go back and you're gonna take a picture of the picture, or you guys just gonna go by memory? Anyone get their phones out and take pictures of the pictures? Alright, let's go back to the first picture. This is on the test. So it's showing you trepe, wave summation, incomplete tetanus, and complete tetanus. Trepe, wave summation, incomplete tetanus, and complete tetanus. So I have some questions about this. All right. You guys have no questions about that? Because I'm going to keep this test open, but just so you guys can't take pictures of my test. I'm going to go down through and tell you the topics that are on it. Now let me give you a disclaimer. I'm telling you the topic that's on it. You have no idea how I'm asking the question. This focuses your studying, but it shouldn't be all you study. When I give you a topic, I mean, a lot of topics are linked, at least to me. So you guys are like, well, not to me. Well, they're linked. So when I give you a topic, don't just presume to know how I asked you about that topic. You might have to know things around that concept as well. All right, first concept is about the lineage of bone cells. So osteoprogenitors becoming osteoblasts, becoming osteocytes, that lineage. It's about the lineage of bone cells. And of course, what those bone cells do for us. Osteoblasts lay down bone, they make osteoid, and osteoclasts digest bone. They break it down. Any questions about bone cell lineage or bone cell functions? Osteoblasts build and osteoclasts break down. Okay. I'll tell you what's on here. The vitamin D3, aka calcitriol story is on here. And I took you way back to the skin chapter. Now don't yell at me and say I already studied that. You did. But the vitamin D comes up again in the bone chapter and it says, you need it for calcium homeostasis. But where does vitamin D3 begin its production? And the answer is in the skin when sunlight hits the skin. So I didn't do it in the same depth and detail as sunlight hits the skin and makes cholecalciferol and that goes to the liver, and et cetera, et cetera. But I certainly want you to know the overview of what calcitriol does for calcium, because we're talking about strong bones. So calcitriol is in here again. All right, let me read a question to you. I'll read the exact question to you. The following are important steps in the process of endochondral ossification. What is the correct order for these events? So I list 5 events. I'm not going to tell you which 5 I list. But I listed 5 events here, and you have to put them in order. So you should go home and look up how endochondral ossification occurs. Any questions about that? Okay, I'm going to read another question for you. Similar. The following are major steps in the process of intramembranous ossification. What is the correct order for these events? So, you need to open your book this weekend and look at intramembranous ossification and know the orders or the stages that occurs in. Now, you don't know how I worded the events, but you got to know what order they go in. Right?
Jeff, that was a question I had because I felt like that wasn't as clear to understand. The way that they explained the intramembranous.
Intramembranous?
Yeah.
Okay, the big thing here is this. They start with mesenchymal cells.
Right. Okay.
So the question I'm answering is, it wasn't as clear, either my lecture or the book, I don't know which one you're talking about, but it wasn't as clear with intramembranous ossification. So let me back off and tell you some big differences. The bones of your skull are formed by intramembranous ossification. The bones of the rest of your body are formed by endochondral ossification. Two different big differences. Intramembranous ossification starts with mesenchyme cells. Endochondral ossification starts with hyaline cartilage. Okay. Now, otherwise, they are similar from here on out. The mesenchyme cells, little islands of mesenchyme cells cluster and they ossify. But because you have these clustered islands of mesenchyme cells that are ossified, you get these spicules. And that's going to be the diploe of the spongy bone. So what happens first, you get the spicules or diploe. And then later on, you remodel that into the compact bone on the outside. Okay, that's intramembranous. For endochondral, what happens is your chondrocytes die. Remember, you're starting with hyaline cartilage. Your chondrocytes die, and the blood vessel that invades the bone, or invades the cartilage in this case, carries some fibroblasts with it. They differentiate into osteoblasts. They ossify that cartilage. Now, the chondrocytes are dead in that area, so they're not going to build any new cartilage. So now you ossified it, and those fibroblasts are osteoblasts, which become osteocytes, and then you remodel that. So the big difference is how you start. Otherwise, cluster of cells, ossify. But how you start is the big difference. Mesenchyme versus hyaline cartilage.
Good, thanks.
Okay. No, I did that without looking at the question because I didn't want to I didn't want to eat it too much. What's the major protein of hyaline cartilage? What are the three fibers of connective tissue? Three protein fibers of connective tissue. I'll give you two of them. Reticulin and elastin. The third one I'm not giving you is more common than both of them. Collagen. Right, collagen. What vitamin do you need to make collagen? Vitamin C. I don't remember doing it in the annotated narrated lecture. It's definitely in your book. You need vitamin C to make proper collagen. Therefore, you need vitamin C to make bones. Especially with endochondral ossification, right? What's the disease of not having vitamin C called? Scurvy. Okay, you guys read that then. Okay. All right. Okay, so I have another concept in here. The mobility of joints. Synarthrotic, amphiarthrotic, and diarthrotic. Synarthrotic, amphiarthrotic, and diarthrotic. Synarthrotic is immovable, amphiarthrotic is slightly movable, and diarthrotic is freely movable. Questions about the mobility of joints? Okay, two fractures I've focused on. So not every single fracture that's in the text is in here, but there's two major fractures I've focused on. One is the distal radial fracture, which is often comminuted, meaning more than one bone piece, and that's called Colles fracture. So Colles fracture is on the test. Now remember, when I say a concept, I just gave you a couple different concepts there. It's called Colles fracture. It's the distal radius, but I said it's often comminuted. So you kind of have to know what comminuted fractures are. So it's not a clean break. It usually has more than one bone fragment. All right. So Colles fracture is on here. The other one is Pott's fracture. Pott's fracture is the distal tib-fib fracture. Tib-fib means tibia and fibula. So it's a distal tib-fib. It's otherwise known as a bimalleolar fracture because you're fracturing the lateral malleolus and the medial malleolus of the tibia and fibula. So Pott's fracture is on here. So how I covered fractures was I hit two famous fractures. In other words. All right, I have some questions in here like this. I'm trying to think of one that is not on here. I have a question like this. Chewing is. Chewing is mandibular depression and mandibular elevation. Another one could be like this. Saying yes is what? Flexion, extension, flexion, extension, flexion, extension. So I have some questions in there where I say, this body movement is, and you choose the answer that matches that movement. Like chewing or saying yes. What's this called? Opposition. That's opposing your thumb, or opposition. So if I said taking your thumb and touching your pinky, that's opposition. All right? All right. Here's the question. I'm going to read it exactly how it's worded. Put the following structures in order from superficial to deep. And I'm talking about muscle. So epimysium, paramysium, endomysium. Okay? And so on and so forth. What's after the endomysium? The myofiber. What's inside the myofiber? Myofibrils. What's inside the myofibrils? Myofilaments. What are the myofilaments made of? Thin filaments are made of actin, troponin, and tropomyosin. Thick filaments are made of myosin. What are they made of? Amino acids. What are they made of? I'm kidding. I'm going further than that. I'm kidding about that. So. All right. So that question, put the following structures in order from superficial to deep. Make sure you know the architecture of a muscle from the epimysium all the way down through. Cool. I like that question. Then I come back to that picture I showed you. I've been through 2 pictures already that I showed you. By the way, let me just show you this picture again, just to tell you. There are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 questions currently to this picture right here. The muscle picture. I'm going to turn it back on. I don't know if I'm going to delete any of them or not, but right now, because I'm at 55 questions, I have to delete 5 questions still. I don't know which ones I'm deleting. But there's 10 questions in this picture right here, as soon as this comes out. I might leave them all. Come on, hurry up, I only got 10 minutes left. I've never kept you guys here the entire time. This is kind of cool. Right there, 10 questions on that picture, currently. All right? There's 10 questions in that picture. Anyone still need it? All right. Next concept. Rigor mortis is a concept on here. Now, just don't look up rigor mortis and say, oh, the cross bridge doesn't break. Well, why doesn't the cross bridge break? Because you're dead. Okay, I get it. You're dead. The cross bridge doesn't break. But why doesn't it break? Because you don't have any ATP, because you're dead. So am I telling you that you need ATP to break the cross bridge? Yeah. So that whole concept about how you break a cross bridge, needing ATP to break a cross bridge is on here. And rigor mortis, of course, is something we see when you can't break cross bridges, because you don't have ATP, because you're dead. All right, I'm going to read one to you. The following is a list of the events that occur during a muscle contraction. What is the correct sequence of these events? You have no idea where I'm starting. Am I starting from the motor neuron firing? I could be. Am I starting from the acetylcholine binding its receptor in the synaptic cleft? I could be. So you have no idea where I'm going to start. So what you guys need to do this weekend is go back to the sequence of events starting from the motor neuron up until the cross bridges form and swivel. And I would know the sequence of these events. All right? I have some questions about acetylcholine and acetylcholinesterase in here. That's the neurotransmitter in the neuromuscular junction. So I have some acetylcholine, acetylcholinesterase questions in here. have some questions about isotonic and isometric contractions. There were two types of isotonic contractions. What were the two types of isotonic contractions? Eccentric and concentric. And isometric contractions is only one type. Isometric is isometric. You don't move the load. All right, I have the picture I showed you. The Cori cycle's in here. And obviously that means that your muscle doing anaerobic metabolism must be in here. Because remember what the Cori cycle is. It's when your muscle makes lactic acid, and then that lactic acid goes to the liver and is converted to glucose and then floats back to the muscle. That's the Cori cycle. That's not in your book. It is in the lecture. It's that picture of the guy's arm like this, and the lactic acid goes down to the liver and comes back to the guy's arm. So the fact that the Cori cycle is in here means you need to know the lactic acid story. You need to know what happens when muscles don't have enough oxygen for their contractions. And that is, they go through fermentation pathway that leads to lactic acid. And then the Cori cycle. All right? Okay. Oh, is there a little bit about it?
Yeah.
Okay. There's a little bit about Cori cycle in the book then. And then I had that picture in the lecture too. There's some smooth muscle questions in here. And towards the end, so there's not a ton of them, but there are. And did everyone get to read the discussion about our smooth muscle discussion online this week, Monday or Tuesday?
Right.
I think Crystal and I were having it, or was it you? Yeah, we were having a discussion Monday or Tuesday. Actually, she posted on Sunday and I responded to on Monday.
Yeah, this is the one that's not in the book. There's no latch.
Yeah, the latch state. So if you didn't read the discussions that we had on Monday, or maybe it was Tuesday, I forget. What I did was I said, here's why a smooth muscle can contract and stay contracted. After the whole story about calcium binding calmodulin and activating myosin light chain kinase and phosphorylating the myosin head and binding and swiveling, after that whole story, there's two things that can happen. One, you can detach the cross bridge and then the phosphatase dephosphorylates. That's a normal occurrence. Two, the phosphatase can dephosphorylate the myosin head before detachment, and that leads to the latch state. Now it stays swiveled, and you don't need any ATP expenditure. So what's important about this latch state, and that was one of Crystal's questions, I said, well, with multi-unit smooth muscles, not much. Because each individual smooth muscle cell acts on its own. But with unitary or single unit smooth muscle, sometimes called visceral smooth muscle, a lot. Because that means a sheet of smooth muscle contracts and stays contracted. So you can contract a whole sheet of smooth muscles and they stay contracted without the expenditure of ATP for a prolonged period of time. Half an hour, hour.
Can you give an example?
Yeah, the peristalsis and gastrointestinal muscle tone.